Hello everyone, my name is Yanying and I'm a PhD student in Mixture Toxicology at the DTU Food. Today I'm gonna tell you a bit about my PhD project and what I'm looking into. So I divided my PhD project into two phases, as you can see on the left and on the right. Currently I am testing androgen and the estrogen receptor activity of pooled real-life samples from the environment, the foods, and the human in vitro using two different reporter gene assays. As you can see, this figure illustrates you the basic principle of how this type of assay work. So we collected samples from, for instance, water and fish, cow milk, human breast milk, human blood, and cord blood. And when the chemicals are entering cell membrane, they can then bind to a receptor to form a ligand receptor complex and translocates into the nucleus and activates the gene that's connected to a luciferase gene. And then the proteins can be translated and measured by a spectrophotometer as they emit luminescent signal. So I am running 2 bioassays out of 20 test battery from our project Panoramics, which is also the project that's funding my PhD. And based on the results from phase 1, we are selecting 3 to 6 bioassays based on their specificity, selectivity, and sensitivity, and of course taking into consideration that we have limited amount of samples to go to phase 2 of the project. And in phase 2, so when we have these extracted chemicals, they are gonna be tested in vitro bioassays, and then our collaborators will analyze the chemical profiling of these 500 human cord blood samples and fractionate the samples and test again in bioassays, and this process will keep going on until we are able to identify the so-called chemical mixture drivers. And of course, the most exciting part of this phase is that we will, in the end, be able to associate in vitro bioassay results and the chemical mixture drivers with health outcomes observed in teenagers who once provided the cord blood samples. And these health outcomes include anogenital distance, ADHD, mini-puberty, and IQ level. So I hope by the end of my PhD project, I'm able to produce a lot of data that can be used to better regulate chemical mixtures. Thank you for listening.